Welcome everyone to this CannX webinar titled The Long and Bumpy Road of Turning Cannabis into Medicine. Thank you everyone for joining us online from the different parts of the world. My name is Pamela and I'm an online education specialist, and we'll be sharing a few technical things with you and some instructions so that you can make the most of this webinar before presenting today's moderator and speakers. So first of all, I wanted to invite you to follow CannX on social media. If you want to tweet or post something regarding this webinar, you can use this hashtag here at the top. And here are all our different social media channels. And I will then post this as well in the chat of the Zoom application, so you will be able to look for these links there as well. So throughout the webinar, you will be able to ask questions to our speakers, and you can do that by clicking on the Q&A section in the toolbar of your Zoom application. And you can also use the chat function if you want to network with your peers, but we are going to focus on the questions that we receive through the Q&A just to make it a bit more organized. So please make sure that if you want to ask something to the speakers today, you use the Q&A section. And we will discuss the questions by the end of today's webinar. So I will also just briefly explain about the certificate of participation. So after the webinar, you will be able to complete a very short survey to receive a certificate of participation for your attendance today. So stay tuned until the end. And without further ado, I will now introduce our moderator, Dr. Yuval Solotov from the Regional Alcohol and Drugs Abuse Research Center of Israel.
Thank you, Pamela, and hello everyone. Welcome to the first webinar of CannX. And wherever you are, I hope you are doing well and keeping yourself safe during this challenging time. Thank you for taking the time to join us. We are very happy and proud to launch this webinar series of CannX, and I'm sure you will be provided with high-level content and gain knowledge on the fascinating field of medical cannabis. My name is Yuval Zolotov, and I will be the moderator of today's webinar. And just before diving into the webinar, I would like to draw your attention to the CannX conference that will be held virtually from the 26th to the 28th of October, later this year. Please go ahead and check our website. We recently added the outline of the program, and as you will see, you will be able to hear interesting lectures from many speakers from around the world. And another important note is that during the conference in October, we will celebrate together the 90th birthday of the conference president, Professor Raphael Meshulam, which I'm sure you're all familiar with. Registration will be open very soon, and I encourage you to take this opportunity and join us for the conference. And now I'm happy to move on to present the speakers. The first speaker, so before presenting the speakers, the title of the webinar is The Long and Bumpy Road of Turning Cannabis into Medicine. And as I'm sure you are all aware, this road of taking cannabis plant material and turning it into a medicine in our times is indeed very long and bumpy, curvy, complicated, challenging, you name it. Today we have two distinguished speakers who are already walking down this road for many years, and they will tell us more about the path that they have chosen to take and share their experiences and their expectations for the future developments. The first speaker is Professor Chinanit Koltai, who is a senior research scientist at the Agricultural Research Organization, also known as Volcani Center here in Israel. Professor Koltai is the leading author of around 90 peer-reviewed publications and of more than 30 books, chapters, and invited reviews. She additionally holds 12 patents. She is the editor of several books and a member of editorial boards in international scientific journals. She also teaches plant development and medical cannabis courses in Bar Ilan University in Israel. Professor Koltai heads the Koltai Lab at Volcani Center in Israel, and the research in her lab is mainly focused on deciphering the entourage effect of cannabis and compounds and specifying active pharmaceutical ingredients, APIs, from cannabis and the medical activity at the physiological and molecular levels in human cells, 3D printed models, and human tissues. The lab combines state-of-the-art chemistry and cellular and molecular biology tools and constantly making new discoveries in the field. The research results are IP protected and they are published in international scientific journals and form a scientific basis for the development of what might become the next generation of cannabis-based medical products, as well as elite cannabis strains. So please welcome Professor Chinanit Koltai. Chinanit, the stage is yours.
Thank you so much. Thank you very much, Yuval and Pamela and CannX. I'm very happy to be here in this webinar and to share with you indeed a bit of what we do in my lab. Excellent. Okay. So, definitely we will start with science. We, all of us, I think, are absolutely sure and we know that cannabis is very helpful and really helps patients that are suffering from different medical indications. No doubt about that. However, really to improve the ability of cannabis to help patients, we feel that the treatment should be better specified. We all know that cannabis produces in the female inflorescence, in the trichomes, hundreds of different compounds, among them about 150 phytocannabinoids, hundreds of flavonoids, hundreds of terpenes. And beyond that, there are hundreds, if not more, of different strains throughout the world. Yet, the medical treatment is based on two compounds only. Indeed, these are the major compounds, but still there are just 2 out of 150 compounds. So this is in the best case scenario where a medical treatment is based on THC and CBD. In some of the cases, it is based on a definition of sativa or indica, which is based on leaf white, but definitely is not based on some real or substantial medical activity. And once we know that there are 500 compounds, it's rather clear to understand why patients are suffering from lack of standardization of cannabis-based products. And this is probably because once we don't know really what is working, then it's very hard to specify compositions that patients should use for different medical conditions. On top of that, we are very much aware of the entourage effect that was defined by Professor Meshulam some years ago as the better activity of several compounds together versus a single compound. So, in order to find the best composition or the optimal composition of active compounds to optimize a treatment of each medical conditions, we certainly face quite a challenge to find out of 500 different compounds and many combinations, what is the best one? And then, as I said, the answer should come to be specific for the treatment of certain medical conditions. To answer this question, we have here in Volcani Center a small but authorized farm for medical cannabis grow for the research purposes only. We do extraction of compounds, we separate these compounds to fractions, and then what we do is we determine the activity of the different fractions and their combinations based on bioassays that we develop with physicians from different hospitals in Israel. The active fractions are going through deep chemical analysis in such a way that we can see up to the last molecule. Based on that, we are able to determine really what is working and answer the research question that we pose about concerning activity. This in collaboration with companies worldwide, we are able to develop new cannabis products. And I will talk about that a bit more very soon. Okay. So we said that we have 500 different compounds. This is basically endless possible matrices, and you can think of cannabis compounds that are present in an extract as a pile of Lego cubes. And what we try to do is to select the right cubes to take in order to treat the patients the best way that we can. It might be that we need 3 yellow cubes and 2 green ones and just 1 red, but this is the best composition to treat a certain patient suffering from a certain medical condition. I will show you one example from of one indication that we are working on, among others. This is a collaboration with Beilinson Hospital in Israel with the Department of Dermatology. In this case, the physicians, the medical doctors, came to us and told us that patients that are suffering from cutaneous T-cell lymphoma, this is a lymphoma that is characterized in the skin by patches of malignant T cells. So patients that use cannabis, and especially those that use it as some kind of a cream, are able to get improvement in their medical conditions. In their medical condition and in the appearance of the actual patches. So it was quite clear that this is something very interesting to work on that definitely might benefit patients. And just to say that the more severe stage of this disease is the Sézary syndrome, where malignant T cells are getting into the bloodstream, and then unfortunately prognosis is not that good. Okay, so what we did is indeed we screened for different fractions. We found 2 fractions in this case that are highly active. We identified the active compounds in these fractions and then combined fractions and active compounds. In this case, what we found is synergy between fractions and between compounds. This, on one hand, allowed the ability to go below microgram per ml concentrations for treatment of cells. And this is definitely something that is very encouraging once you consider treatment of patients. That's on the one hand. And on the other hand, what it led us to the thinking that this actually might be the entourage effect, this synergistic interaction between cannabis compounds. Okay, so once we knew what compounds are working together, we were able to define the active pharmaceutical ingredients, or in this case, active plant ingredients. And this is just an example. And for treatment of a certain medical indication, you need such and such amount of CBG and that amount of CBN and that of CBCN, and so on and so on. Aside from knowing what the right composition is, which is very important, we were also very much intrigued about this process of synergy between compounds. And to better examine this, we went deeper into the biology, and we asked what is happening in the human cells once they see the synergistic compounds acting together versus just individual compounds. What we found is first that only the synergistic interaction leads to a formation or induction of apoptosis, programmed cell death in these cancer cells. We did the RNA sequencing, and we were able to find around 3,000 genes in these cells that are altered, either induced or reduced due to the synergistic interaction. This led us to the identification of signaling pathways in these cells that are being altered by the combination of compounds from cannabis. And then we started to talk not just on combination of compounds, but rather on genetic pathways that are being affected by this treatment in the malignant cells. Interestingly, some of these pathways are known targeted for chemotherapy. But I think the most exciting finding that we had is the findings of activity on cells that were taken from patients, from cesarean patients, where the T cells, the malignant T cells, are already in the blood. In collaboration with Belinson, we received these cells, and with them we checked together, and we determined that we are able to kill these malignant cells and indeed induce apoptosis in these cells by combination of fractions, and we were managed to do that significantly better than the effect on non-malignant cells. So we have some specificity, not complete specificity, but some specificity to cancer cells. So if we quickly summarize what we talked until now, we started with many strains and compounds and endless possibilities. Using bioassays and chemical analysis, we were able to define fractions and then compounds that are working synergistically. We looked a little bit into the targets and mode of action of these compounds, and now we are in stages of development of these API formulation into drugs, into cannabis-based drugs. And we think that this is the new generation of cannabis-based products, where products would be based on specific composition of API. Definitely, this is a long and bumpy road, and I fully agree with the name of the webinar here. But this is a road that we are going in, and hopefully hope for the best. This road necessitates a very deep interaction between our world, the academic world, and the business world. And this is certainly something that we experience here. All our projects are being in very close collaboration with business. And this, has to be done, has to be, because we are both doing research and development. But we also interface with agriculture. And once we know what API is needed for the treatment of a certain medical condition, then we are able to develop here elite strains that are specified for the treatment of a certain medical condition. And if I look into the future, I think This is how I see it. I see perhaps lines or plots of cannabis strains that are the best ones for the treatment of a certain medical indication. We asked additional questions. I will not go into too much details here, but we asked about the entourage effect and the interaction between phytocannabinoids in between themselves, which might be called intra-entourage effect, and then between phytocannabinoids and terpenes, which might be referred to as inter-entourage effect. And, you know, there's quite a debate if terpenes are really needed, are really important for the activity of cannabis. And to check that, what we did is, first of all, we defined in about 70 different strains, we defined the distribution of cannabinoids versus terpenoids. We found correlations between the two groups of compounds. We separate the terpenes and were able to show that indeed with terpenes we can enhance specifically the activity of certain phytocannabinoids. This led us to some insight into the strains of cannabis that we have today. What we suggest is that cannabis domestication that was done by humans for ritual or for medical care led to the strains that we see today. And in these strains, the distribution of phytocannabinoids and terpenes is not just a coincidence. Rather, this is derived from breeding and selection by men for thousands of years into these strains that have entourage between phytocannabinoids and between themselves and between phytocannabinoids and terpenes. And what we think that by utilizing this entourage, we're able first to optimize the cannabis-based treatment and perhaps also to improve taxonomy of cannabis chemicals. And with that, I will finalize with the acknowledgements to my collaborators. First of all, people from my lab, Dr. Dvori Namdar and Richard Lev, and our dear students, the Israeli Gene Bank, and our collaborators from the different hospitals and medical centers we're working with, and Bellinson, as I mentioned in this case. And I thank you.
Thank you very much, Chinanit, for your interesting talk. And if anyone has questions for Chinanit, please keep them for the Q&A session at the end of the webinar. And as Pamela told you, use the Q&A function in Zoom to send us your questions. And I'm now happy to present our second speaker, Dr. Fabricio Pamplona from Brazil. Dr. Pamplona is a pharmacist and psychopharmacologist who is specialized in the modulation of the endocannabinoid system and the therapeutic potential of cannabis with 15 years of experience in science and technological business. His career path includes a position as guest researcher in the Max Planck Institute of Psychiatry in Germany and as life science specialist in Lionbridge in the USA. He's an international speaker with participations in the Gordon Research Conference, International Cannabinoid Research Society, TEDx, South by Southwest, and CanMed in the Harvard Medical School, among others. On top of his significant scientific careers, he has relevant entrepreneurship experience, having founded 2 tech startups and served as mentor for a number of other ones. His major scientific contribution so far has been published in the Proceedings of the National Academy of Science and highlighted as a promising medical product in Nature Reviews Drug Discovery. As a following step in his research, he is working on a diagnostic tool for the endocannabinoid deficiency syndrome in Alzheimer's disease. His last position was R&D Director of Entourage Phytolab, a pharmaceutical startup company dedicated to the development of cannabis-based medicines. He has authored 50 articles published in international peer-reviewed journals with over 1,800 citations, and currently serves as Head of Science at Proprium Health Technology and Science, which is based in Brazil. Please welcome Dr. Fabricio Pamplona. Fabricio, please.
Thank you, Val, for the kind words, and congratulations, Dr. Hinanit, for the excellent presentation. It's the entourage effect and the idea, the concept that the molecules interact among them synergistically. It's a very exciting frontier of research. I also dedicate part of my career on this when it comes to allosteric modulation and description of the physiological interactions between molecules, which ultimately made me step into product development, as Yuval or Tubi for the friends just mentioned, that is being used for potential diagnostic of the endocannabinoid deficiency syndrome that I'm gonna present in the main event. But today I restricted the webinar to just part of the presentation and part of the developments that I'm doing at Proprium. Proprium is the startup of precision medicine. So we use genetic tools, biochemistry tools, and interviews, clinical scales, all these sort of things, trying to create a different experience about around the use of cannabis. But not only that, we also create a new experience around nutrition, around fitness, around use of medicines and personalized treatment in general. But what I'm going to present today is the concept of using genetic profiling. It's an actual product that is already being sold. We developed together with a genetic company from Portugal, where Proprium also has offices and labs, that helps the prescription of cannabis-based products or cannabinoid products. And I think it's more straightforward or geared towards the clinical prescription and the utility of the method and the concept, but for sure we can dive deeper into the science behind it during the Q&A. So feel free to ask questions by the end. A little background on myself. To be already said, but I'm a pharmacologist. I just wanted to do this disclaimer. I'm not a prescriptor. I'm not a doctor, but I'm a really experienced cannabinoid pharmacologist since the beginning, since beginning of my career, 2003, so 17 years. I passed through, during my PhD, through the Max Planck Institute of Psychiatry. I did part of the PhD there in Germany, and then in one private institute in Brazil, doing clinical research on diagnostic tools that ultimately gave me the background to be where I am. And after that, 2 different startups, Entourage, that now you know where the name comes from, and it was devoted to use standardized cannabis strains, initially out of bedrocan into pharmaceutical development. So the strategy is pretty much aligned to what Dr. Hinanit just presented, and I really believe and trust and know that this is a very good strategy in the long term. However, at the moment, what most of the patients have available is mostly THC, CBD, even on the purified form, or in a certain form of standardized extract, even though it often doesn't come from a standardized strain. Sorry for that. And at the moment, I'm co-founder and CSO of Proprium, and where I'm developing tools that help prescription of cannabis and other products based on the patient biology. So I'd say this is the time that the highlight that I want to make is that everyone is talking and trying to understand the biology of the cannabis, which is truly important. But on the other side, we also need to understand the biology of the patient that is taking the cannabis or the cannabinoids, because we talk about the entourage effect, that's true, but also both organisms, if you will, need to also synergize, and also we need to find what is the best composition for the best patient biology, if you will. So just taking in this figure, like in a heterogeneous population, several patients with a given condition, say chronic pain, they may be very, very different response to the same treatment. Theoretically, some of them may have good response, some of them may have no response, some of them may even have bad side effects. And there is a ton, over 100 papers on pharmacogenomics of cannabis already published. So there's a lot of scientific work done on the characterization of genetic variations that lead to differential response to the cannabis effects on different people. That's the groundstone for all this work. And so that's, let's say, the concept behind everything we did, and the strategy for the product is, given that in a certain population we have different people with different, let's say, DNA, we use profiling of certain targets. I'm going to show you in a minute what is the panel we chose and why. But we can use different profiles and use this strategy to give recommendations or make actionable results, allow people to take actions or to make better decisions based on the results of the genetic test. So what we know from the frequency of the sequencing that we've done so far. So most of the people, they have a normal metabolism for THC or CBD, like in general, and they don't have really, really high risk for adverse events. That's the majority of the people. Some of them have normal, let's say, THC metabolism, but also some risk for adverse events. And very few people, they have either higher risk for let's say, THC overdose because of inhibited metabolism, and risk of developing serious adverse events. For instance, the psychosis, which is kind of the ghost in this therapeutic area, and where most of the clinicians say, no, I don't want to use cannabis because I don't want to put my patients on the risk of developing psychosis, or even worse, they say schizophrenia, which is way down the road. So what we want to do with this tool is getting rid of this fear and make all the decisions more actionable, more reasonable, more rational. And I think that understanding the biology of the patient may well contribute to this. So why should one take the test? We have the idea that anyone taking cannabinoids should, before take a test like this. And I go even deeper. Anyone being born should understand its genetics, it's our nutrition, it's our body type, response to treatments and things like that, because this is an act of self-knowledge. It's very convenient. Just get a sample inside your cheeks. It's so-called saliva harvest. It's not a saliva, it's the cells, because we need the DNA. And after genotyping, we run the data, the raw genomic data, through a proprietary algorithm that identifies clinically relevant SNPs, so polymorphisms, natural variations of the genome that leads to different effects. And after that, we can then generate and identify different profiles of patients to make recommendations. And I must say that this test is indicated for any individual in any age range. We have cannabis patients which are elders. We have cannabis patients which are regular adults, but we also have children. Anyone can take it. We like to understand it as a journey of self-discovery that improves your ability to make better decisions, and your clinician as well. And it can be useful even before you develop any pathology, just to be on the safe side, to understand, if you will, or if you perhaps have genetic information that you probably may develop a disease, or your family has a certain tendency for a disease, you may decide to take tests like this to be ready for that, to be prepared for that. And if cannabis is part of this, let's say on multiple sclerosis, you may well understand whether or not this treatment is suitable for you, whether or not this is even an option. This is the panel we decide, or I designed it together with our partners. So it's a full panel with several different targets, trying to understand the metabolism of THC, CBD, and the metabolism and the metabolites of them. Of course, a very common question here is, why don't you have the metabolism for different cannabinoids as well, hair cannabinoids, things like that? Because the science is not ready yet. That's basically the answer. We can improve and add new targets as soon as we have more evidence from the scientific side. And I must highlight that in the metabolism, we are the only product of this field that have metabolism of phase 1, phase 2, plus the glycoprotein P, which is important for extrusion of the central nervous system. So we have the cytochromes for degradation. We have the UGTs enzymes for the excretion, clearance of this drugs from the body, and also the ABCB1 gene for glycoprotein P for extrusion of the CNS, which is very, very important when it comes to cannabinoids, because you may well have someone with good ability to metabolize the cannabinoids in the liver, but it might still lead to a higher concentration in the brain because this person has a SNP in the ABCB1 or in the glycoprotein P, so the relative level of THC, for instance, in the brain is higher than in a normal person. That's the kind of thing that we can say after doing the test. When it comes to adverse effects, we have different genes, and they indicate adverse effects like anxiety, like tendency to psychosis, like tendency to addiction, and the cognitive effects, and the direct link with the cannabis, the addiction, typical addiction of the cannabis. It's called the marijuana problem scale. It's a specific clinical scale for evaluation of patients in this field. And also addiction in general, we have many more targets just because there are more science on this field than on the others. But I consider that this is an evolving field of research, and we can add many more targets now that the whole world is looking at cannabis via medicine or therapeutic perspective, not how we used to be in the conference before, that everyone researching cannabis was looking for cognitive deficits or problems in addiction and so on. So on the right-hand side, you'll see the table, how it goes, how the information that we have, like the raw genetic information, the SNPs, position, things like that. Of course, you as a clinician may have this in the report as well, but we think that one of the most interesting things that we did is to translate that information into an information that is actionable, really easy to understand, even for the patient, and ready to go, to make a decision. So these are the fields, like the sections of the report, have metabolism of THC, CBD, and how these treatments may affect your cognition, psychological and behavioral states like anxiety and tendency to psychosis, impact on chronic use of phytocannabinoids, which is a better way of saying addiction, and the impact on our daily life, which is another way that we use that we prefer to use to refer to the marijuana problem scale. So this is how we see internally. We have THC and the metabolites here as one example, but of course the same goes for CBD. And we have an N, like the neutral metabolism, or partial reduce, very reduced on the left-hand side. On the right-hand side, partially improved or improved. So that's how we indicate the combination of those 7 targets related to metabolism. We either say, in general, our algorithm decides that you have even reduced or increased metabolism for every compound. And for THC, we know that different metabolites impact on the administration route. So we differentiate between the 11 and hydroxy THC, which is important and is an active metabolite, which is even more psychoactive than THC. that is important for treatments through the oral route, and also the 11-carboxy THC, which is important for the patients taking inhaler cannabis. We recommend initially dose for THC, a maximum daily dose for the initial titration phase. One of the aims of our report is to speed up the titration process. That is where we acknowledge that is always important, particularly for cannabis naive patients, but we want to speed it up. We understand that is a process of trial and error between the clinician and the patient, but we need to make it faster and better. So this is a report of the other aspects that we measure. This is the very first page. They have a summary of everything. Of course, you have several details and information on every SNP that is evaluated. But one of the main things of the report is that we generate the index and show the information all in one bar. So it's very actionable. And this bar, or behind this bar, you have certain weights depending on the SNP and depending on the source of the SNP, the science behind it, that takes into consideration the effect size of the research, how many patients the researcher used it to take that conclusion, what's the significance of that conclusion, is it linked to any particular ethnic variation, and then of course we take into consideration whether or not the patient is a part of that group, but also very, very important, the frequency of the distribution. So in the population. So if you see index bar like this, and you are around the 50%, so in the half, let's say, of the index, it means that you are intermediate, which is another word to say normal. You have a tendency to that adverse effects, which is equal to a normal distribution of the population. If you're below, you are eventually at lower risk, and if you're higher, you are on the higher risk. That's how we do. And the second page is the recommendation of products based on your biology. So I just took one patient as an example. So here you see that this patient can take mid-range THC, which is the orange, low-range THC, and equilibrated balance, one-to-one THC CBD products, higher CBD than THC, so very trace THC or hemp products, it's in yellow, and the nude or rose, it's pure CBD products. So all in all, I want to conclude this part of the presentation saying that genetic profiling is important for cannabis prescription because it improves decision making for the clinician and the patient. It optimizes treatment benefits, helps avoiding adverse events, and saves time and money in dose titration. If you're interested about these products, this is the website ipropium.com, and the name of the product is My Cannabis Code. Thank you all for the attention. It was a pleasure. I'm ready to take some questions. And I guess you just hear my kids entering the room. Sorry for that.
Thank you very much, Fabrizio, for this interesting and inspiring presentation. We have quite a few questions, so I apologize in advance in case we are not able to address all of them. First question to either Hiranit or Fabrizio, whoever wants to answer, from Professor Marwan Svetsun. Oh, sorry, I'm probably not pronouncing this right, from Canada. He's asking, how does plant cannabis impact the natural endocannabinoid system, the central nerve system, GI tract, immune system? Question mark.
Thank you very much for your question, Professor. I think this is a very important question. And I think that as for now, we definitely cannot answer it fully and not even answer it much. I believe that finding the actual changes that we infer on the endocannabinoid system is definitely something that we should explore more. We don't know much.
I want to just contribute. I found this very interesting paper of a colleague of yours, like the David Murray, recently published something about the cannabinoidomics, as they are calling. And I find it a very, very interesting approach to answer this kind of question. I agree that we are not ready yet, but the idea of, evaluating the full cannabinoidomic, sorry, and having the different targets of the endocannabinoid system, the different molecules, and start systematically treating with different cannabinoids may lead us to be able to answer this question quite recently. And I was very glad to see this paper recently. I need to even go deeper in that. It was published last week, I guess.
Okay, thank you. Now another question is quite a general one, but maybe you can give some insight about it. Do you have ideas already about which varieties of cannabis are fit for which treatment?
At least from the work that we do, as I said, we are starting to understand what compounds and what compositions of compounds are the best one for treatments of different medical conditions. Based on that, what we have started doing here is to select for strains that can be designated for the treatment of certain medical conditions. So definitely we see that as the continuation of our work and the part of our mission to be able to specify strains to different medical indications. This is an ongoing work. We start to have this kind of strains, and this is something that we are working on, definitely.
I'd be really, really glad to cross this concept with what we are doing, because at the moment, although of course I understand and believe in the entourage effect and all this, I did part of my research. Our study is restricted to the CBD and THC because that's what science allows. But since we have the tools now, we can cross it and use the same patients for your treatments or even the animals and have them genotyping, and start to make scientifically consistent genetic profiling linked to strains that some products are claimed to have, but it's totally far-fetched, unfortunately. But if you have standardized strains, that is a powerful tool for the patients. That's all I have in mind, always to empower the relation of clinicians and the patients, and particularly in countries like Israel, that the patients actually have access to strains, to proper medical strains with proper quality. That's powerful idea.
Okay, thank you. And then next question is for you, Dr. Pamplona, from Eduardo. He's a pharmacist working both in Michigan, in the US, and in Peru. And he writes that he realizes that some patients are susceptible to the medicinal properties of cannabis, but still they are not able to experience the psychoactive effect of THC. And he asks, what are your comments in this case from a genetic perspective?
Mm-hmm. It's a very interesting comment. First, it depends on the product. Some clinicians refer that they are not sure about the product composition, which is unfortunately true in some countries that doesn't have proper regulation. But from a genomic perspective, I'd say that it might well be that these patients have an increased metabolism and therefore will require higher doses. That's one possibility. I never really heard of anyone that it's, let's say, that cannot experience the psychoactive effects, but I heard of people that needs to take like 100 milligrams of THC to be able to feel it. So it might well be the case.
Okay, thank you. Another question from an anonymous attendee is asking how can one recommend an initial titration with confidence if different products can have different absorption levels, even if we consider the oral consumption?
Absolutely. And of course, Dr. Hinanit can complement on this, but my perspective is that it's the duty of the manufacturer to inform about the proper absorption and administration of the products. And some of them even claim, let's say, nano compositions and things like that without any decent clinical data to support that. And I would say it's the responsibility of the manufacturer.
Okay, thank you. So we have basically run out of time, and I want to finalize today's webinar and to thank you once again, Professor Coltai and Dr. Pamplona, for your interesting talks. And both Hinanit and Fabrizio will also be speakers in our upcoming CannX conference in October. And we hope to see all of you joining us there for what promises to be one of this year's most prominent events in the medical cannabis community, with many interesting speakers and network opportunities. Thank you again for joining us. Please feel free to email us to this email address if you have any questions. Keep safe wherever you are, and see you in our next webinar. Thank you very much.
Thank you. Bye-bye.
Ciao. Thank you for the opportunity. It was great.